when they want to. Thank you so much for your help. Bye. Okay, so this afternoon session will be dedicated to fungal and bacterial culture, so fungal and to parasites of phenophagorates with the algae. Ask Pietro and Albert to come here. So you know Albert is WC1 leader. He's a researcher at the Marine Science Institute in Barcelona. His research has always been related to the study of unknown microbial diversity in marine waters. This knowledge gained has allowed him to work with harmful algal blooms, parasitic interaction, and fungal communities. He is now focused on unveiling the protein diversity in benthic coastal settings. So Pietro, as you have heard yesterday morning, won a fellowship within the GOST section. It's a fellowship.
It's a long-term scientific mission.
Long-term scientific mission through ParAqua. That, as you know, is a possibility that it's open, still open, more or less, for people to. Okay. What is it there? It went running and it's not back. Okay. So there are still possibility for people to apply to this short-term mobility. So look at the ParAqua website and look for people that can host you and do some amazing research together as they did. Pietro is also a researcher here at the CNR IRSA, and she won a Marie Curie with the amazing score, and they will be working. Maybe you can tell what you will be working.
Yeah, I will work on zoosporic parasites of phytoplankton, and the disease is mediated by the phytoplankton microbiota, using mainly metabolic coding to look at the bacterial diversity associated to healthy or diseased phytoplankton, and also bioassays to test whether adding bacteria that were mostly represented in phytoplankton cultures that are resistant to those parasites would provide resistance in susceptible phytoplankton hosts. So essentially, yes, maybe I didn't explain it well, but the disease dynamics mediated by the microbiota and how we can use the information from environmental diversity to provide resistance to new cultures and to phytoplankton in aquaculture essentially.
And his mentor would be Esther, that you have heard about from this morning, and then you will also do a part with Silke, no?
Yes.
Okay.
Yeah.
So now we divide in two groups, 2 hours and 2 hours. One group will go with them. So the second group will not have the introduction speech. And the other group will stay here. So you're going to look at the phenophagorates parasites in the other lab, and here fungal and bacteria cultivation. Okay? So for my participants, we just have the lecture on bacterial and fungal culture here, because in the other lab we don't have the connection. Okay? So you can grab the people that want to come with you.
So we take first 9 or 10.
So we are
trainers as well.
Participants from 4 participants, 9, then the teacher can decide. Like the teacher want to participate, just split equally. I don't know if Lucas, you want to go to what you want to say.
I think we can take this. Isabella, you want to stay here?
Yeah.
So we take.
Yeah.
Anyway,
we repeat. We will repeat. We will repeat. In 2 hours, we will repeat the test. So you will know. Okay.
We have all the teacher brought.
All the teachers from us.
No? Non può. No, sono I ragazzi più grandi. Vi devo raccontare anche.
No, so, 9 formal participants should go and 9 formal participants should stay.
We did need one.
No, but
No, it's okay. It's okay. It's okay. Like this. Oh, okay. Okay.
Just like breakfast.
Because Laura, we are not staying for the second part because we need to do samples for fresh samples of that tomorrow.
So where is Andy?
He just went to wash them.
Ok, so yeah, no, siamo fatte di pictures e poi andrà correndo e la va correndo. Ok.
Okay.
And all this stays in the registration plan.
Forever.
Discussion.
Forever. Okay. Nice to, I will especially talk with the people at home because in the room there are people that really knows what we're going to tell, but anyway, I'm very happy anyway. So people involved in this real collaboration activities. It's are myself, of course, Manuela, Laura, of course, you know already, Pietro went around. I'm really happy to introduce to you Zacarias Fresno Lopez, who is a fellow student, yes,
Well, that's better.
yeah, research fellow, okay,
That's easy, my speak.
at the IRSA, and today he will talk about, of course, bacteria, not of course, without of course, and Selene, she has already been mentioned. She's on maternity leave because she works on fungi. I like to introduce myself. I'm a microbial ecologist since an old senior microbial ecologist.
You're never too young to get
Yeah. Yeah, senior scientist. So for a long time, I haven't been working really in science. I've been working especially with microbial co. So I really focus on the development of especially educational and training activities. That's why I'm still here. But now I work in the construction, let's say not really construction, but the development of economic observatory in the Adriatic Sea. So in the Adriatic Sea, in the CNR IRBIM in Ancona, we have they had the facilities that were really like a sea mark that was collecting, from whom we were collecting daily physical and chemical parameters. And then now we implemented, during this year, we implemented the omics sampling. So we developed a standard operating procedure to take a monthly sample and to perform, on which to perform omics activity. But today, the topic is cultivating and preserving microbes. So, and I think the first consideration why you decide to cultivate bacteria or fungi is to know which type of bacteria you want to cultivate. And the first thing two questions you have to ask yourself. Which kind of, from how they are producing energy and from which type of carbon do they use? So this workflow really meant for you to decide where you have to go, because cultivating bacteria, especially if you work with chemolidotrophs, can be like seeing pig flying. So it can be something you will never be able to manage in your whole entire career. So what we have been doing today was working on phototrophy. So phototrophic organisms, the one that they use light for energy. And now today we will see some chemiotrophy. So living organisms that they use chemical energy, and they produce ATP from this. And then you can also choose between organic carbon or inorganic carbon. Where to put fungi in this? Yeah?
So, what?
Where to put fungi in this? Exactly. So there is space also for fungi. One important thing that I will tell afterward, because I will just talk a little bit about chemolithotrophic organisms, especially ammonia oxidizers, so a really subgroup of chemolithotrophic organisms. It's what also Esther told today. It's a growth factor. So especially if you work with bacteria and chemolithotrophic, one aspect to really take into mind in your mind is what you put in your medium. And natural medium or sterile water, or artificial, sterilized marine water or sterilized fresh water hardly work for cultivating some type of bacteria. So you really need to make your synthetic medium. And now Zach will explain a little bit the basis of the consideration we have to do for medium, and then I'll be back, and then we do with fungi.
I keep water close. Okay, yeah. So I'm Zacharias, as I said, I'm a research fellow here in the Institute. And I am currently working in a project in which I study the bacteria and microbial community in general of the Adriatic Sea, and I'm also working. I've been working a lot lately on bacterial cultivation, so that's why I'm talking about the basics of bacterial cultivation, and mainly focus on the heterotrophic bacteria. We see from the diagram that Manuela showed that those that use organic carbon as organic soil, they basically break the organic matter. They use organic carbon to as a carbon source. Why the plant aesthetics? Yeah, because I like it. Yeah. So yeah, they can be important in many of our studies because they are present basically everywhere where there is organic matter, water, food, soil, vegetation. Also, they are within organisms. Our microbiota is heterotrophic bacteria, and they are important indicators of water quality, pollution, and also all the human pathogens are heterotrophic. So that's why they are interesting to look at. So in order to cultivate heterotrophic bacteria, or in general, we need to know the kinds of media. There are different media based on consistency, like we have liquid media or broths that you don't put any agar, they are just the normal media with water. And as we can see in the picture, the one in the middle, the bacteria grow uniformly. Then we have solid media, the agar in petri dishes that we usually see. And they are good for looking at the morphology of the bacterial colonies, pigmentation, etc. Also, there is a semi-solid media that is normal media with a lower quantity of agar, and they are usually good for looking at motility of bacteria. And then, based on the function, there are general purpose media. There are media with nutrients, in which basically you don't focus on any kind of or any specific organism. You just, for example, the nutrient agar is rich in nutrients, so you introduce your sample and bacteria grow there. Also for fungi, the PDA, for example. Yeah, this slide has the first one is an example of for bacteria, and the second one is an example for fungi. Then we, for example, we also have low nutrient media, in which basically, if there is a bacteria over competing some in your community, putting lower nutrient may help these bacteria that are usually over competing. Then, and this is how they usually look like. There's normal colonies. We see normal colony growth. Then we have selected media. Sorry, this is not. But that are media that focus on specific organisms, the different specific metabolisms or specific genera or species. Which these are the ones these are the only ones growing that media. Then we also have the differential media, like for example, the blood agar, in which there are different chemical compounds that allow the growth of bacteria and differently, or either they react with the media and they grow in different colors, or even the media looks different, they have different kind of growth. As we can see here, this is in blood agar. It's easy to see, for example, when you grow for alpha, beta, and gamma hemolysis, each of them looks different. Then a super group of differential media, there is chromogenic media that are the plates that we have here. For example, the TBX agar, they have a specific
Chromophore.
That the bacteria reduce and
Hydrolyze.
Sorry?
They hydrolyze and then
Yes.
they change the pH and then they have some pH indicator or that they change.
So that you basically, for example, the TBX agar is specific for Escherichia coli, because Escherichia coli has the beta glucosidase, and it grows forming blue colors. So here you can see a normal culture of bacteria, and you can see the Escherichia coli. Then, knowing all these kind of media, how to select them, you have to consider first the type of sample you have, if it comes from water, if it is a clinical sample, what bacteria or fungi you are focusing. Because if you want to grow just a specific organism, you use the differential media or selective media. And or if you want to look at pollution, so you can also look at chromogenic media. Then you have to consider the different growth conditions, the temperature, the pH, also the aerobic conditions in which your bacteria, the one you want to focus or the one you want to look at, grows. Then, the purpose, basically, what you want to do with your sample. Then, in order to cultivate, there are different techniques. You have to take all of this account, and also the different techniques of cultivation, how you introduce the bacteria in your media. Like there is a streaking, for example, in which you just basically have a loop full of your microorganism, and you spread it in the plate like this. And so this is mainly for the purpose of this is mainly to isolate colonies. Then, for example, for counting, it's also good the pour plate method, in which you introduce your sample, and then after that, the agar, and you let the agar solidify and the media solidify with the sample inside, and it grows, forming colonies. You usually need to dilute this before, but I will talk about it later. And also the spread plate. Like you introduce in solid media your sample and spread it homogeneously. You also have to take into account which temperature your bacteria will grow, 37 degrees for human pathogens, and you have different temperatures. Then, it's all of this. Then some problems during cultivation, I think you mostly learn through experience, and are contamination. Like you have bacteria growing that shouldn't be there. There is not the bacteria you're looking at or you want to look at. There is someone waiting. So
there is someone waiting in the room.
You cannot read.
Yep.
Even from that.
Okay. Okay. So contamination can happen for a problem of sterilization, like there is pollution maybe in the lab, in the fume put you are working, or maybe your air. And it can be detected through, you can see that if you know how your bacteria grows, or even if you see different colony sizes, you see different consistency, color, And this is mainly, I mean, this is, I think experience is what shows this. For example, I was talking with Manu and she told me to tell this. I had a problem. I'm working with Escherichia coli and I'm isolating Escherichia coli. And at some point of the whole lab process, I found that in my agar,
We have a new Fleming
yes,
here.
that in my agar there was the normal colony that I was seeing. It was kind of slimy and was always uniform, but at some point there was a white dense mass inside that. So that was already a proof of pollution. But then I tried to isolate it to see what it was. And when I managed to isolate it, I also started noticing that it in the liquid media, it had different turbidity. It was very, very less turbid. Also, it didn't smell. Like the nose, Esther, remember she told me that the nose, when you start recognizing bacteria by smell is when you become a microbiologist.
That's true.
Yeah, and it smells different. So then you start noticing pollution. So how to prevent this?
But you had to tell what was the
Yeah, I will tell you later.
Ah, okay.
Yeah, I keep this story aside 1 second.
This is fans.
Yes. At the end, we knew what it was, but yeah, so basically you have to be very sterile, sterilize everything you use, be careful not breathing on the plate, using controls, negative controls all the time, and repeating purification. And sometimes with using pollution, you have to go back and keep going until you have a clean evidence. So in order to do this, how to work the most aseptic way when you are working with a bunsen, with a flame, this creates a sterile area around the flame. So you usually want to work close to this area. Like here, for example, also, I'm not opening the plate until I want to go with the loop. So I'm working close to the flame and I am reducing the time of contact with the media and the air, basically. Then sterilizing the loop every time you use it. I use alcohol and then flame it. But also, if you do this, be careful, wait a little bit, because if it is too hot, you kill the bacteria. So,
Can I say one please?
sorry?
Work with gloves.
Yes.
And
Yeah, it's
yes. So just one thing, since for insurance reason, we cannot use flames or access to the laboratory, that's why we made the video. So you can see the full procedure, you will have the video and you can see it directly.
Yeah. And then also the way you manipulate the Petri dishes. So every time I'm working with them out of the hood, always wearing gloves, not opening it, and I also have a paraffin around it to avoid the air or humidity getting in. And yes, this is the main way to work. Most aseptic. It is the most aseptic way. Then once you have managed to isolate your bacteria and yeah. How can I confirm that this is my bacteria? So basically, you go back to the molecular analysis, and but part of the chromogenic confirmation and so on, you still sometimes need this double check. So for bacteria and also for fungi, there are specific genes that are very conserved. That are highly conserved through the whole kingdom. Like for example, 16S in bacteria, I don't remember in fungi. Well, the 21 ITS in fungi. Yeah, so they are very stable genes, but with some variable regions. So so you usually look at those regions, and you basically just extract the DNA, and through PCR amplify the region you are interested in looking at, and so you at the end have many times that same region, and you can send to sequence. And through single sequencing that I will not explain now, that it will be in the materials later. You can double check that you have an isolated strain. And then now.
No, wait,
Yeah.
can I add something?
Yes.
Okay. So Zach put like to move to the practical part. Zach put the video, but we decided for now to not show it. But in case we can come back later if we have time. But you will have it in the presentation. And for fungi, I know which one of you is working on fungi. So basically, IPS is the barcode region. But if you want to arrive at species level for a lot of fungi that you will look at today, I prepare a table for the markers that I use. That are basically, I use the CBS, the Center that now is Westerdijk Institute in the Netherlands. I follow their publications to choose the best marker to identify the species level. I will make a table and I will provide it to you in the material, as well as the two methods for extracting DNA from fungi. One is CTAB method and one a modified DNA plant kit for extraction of fungi. And I know that Isabel tomorrow you will talk a lot about the importance of the barcode of fungi, so I will not be long because you will see it tomorrow. But just you will have these three resources in the materials, okay?
I just wanted to add, then with my story of the random strain.
This is
The fact is that at the end I isolated the strain and I extracted DNA and sent to sequence. And when I looked at it, I did the blast with the FASTA file. What we saw is that it was Streptococcus hemolyticus. So it's not a very nice strain to have. So it was basically my problem because at the point I was working with that with those plates, I was sick. That means that I was breathing on the plate. Yes.
No.
No, like we use here environmental strains that are not BSL risky. So basically, it happens that when there is a pathogenic strains, you just throw everything away and sterilize. Even fungal culture, we don't want to work on human pathogen, basically. So we will not keep these conditions. But of course, if you're working on an environmental sample, it happens always. So this is the case.
Yeah, and also, I mean, we knew that it was pollution, that it wasn't meant to be there, and it wasn't in my sample, because Streptococcus is a gram-positive strain, and the media I was using was for gram-negative at the beginning, so it's impossible that they grew into gram-negative. So that's why we just threw it away. It wasn't in my sample. It wasn't interesting. It was something that came afterwards.
You had some antibiotics today.
Yes.
I didn't know. I mean, I wasn't that bad. Actually,
How do you call it? 0 kilometer homemade bacteria.
Contamination. Yeah.
we banned everything.
Yeah.
We sterilize even back.
Okay. Just because, of course, when you work with bacteria, generally you buy the medium, no? You have this sort of quite sometimes expensive medium.
This was very cheap.
Yeah, this is actually from for fungi. Anyway, if you work or if you want to isolate chemolithotroph, you really to consider, keep in mind that you are working with bacteria that are using an inorganic, so a chemical reaction for producing energy, and they are using an inorganic carbon source. So I'm going to give you the example of the medium for ammonia oxidizers. Ammonia oxidizer, as the name say, oxidize ammonia. So in their media, there should be something with ammonium. The best salt generally used for ammonium is either ammonium chloride or ammonium sulfate. Okay, so this has to be present. Then, of course, you have something like a phosphate component buffering the system. And then you have one key element in cultivating chemolithotroph is a pH indicator. Crescent red is a pH indicator. This receipt has been taken from the DSMZ. It's the medium 5083. Now they really implemented the page for the media, and tomorrow you will meet a person who is working at this at the DSMZ. And if you navigate in the, I don't know if it happens to you to navigate the collection, they have an amazing amount of information about the medium and also tips. So it's very useful. DSMZ. So this is the official medium, but in my past, I've been working in the laboratory in which we're cultivating ammonium oxidizer, and they have a very huge collection. So the media is slightly different. For example, this is the collection. You see this bluish culture. This is the pH indicator. As soon as ammonium oxidizers are growing, they are reducing the system because they oxidize the ammonium. So the pH turn yellow. The medium turn yellow. And this old lady who was maintaining the collection, she was not using this Creso red, but she was using another pH indicator, this bromo, this is impossible to say in, yeah. Sorry, I have to read it again.
Bromotimol blue.
Bromotimol blue, that I think you're using also, no? No. Okay, bromotimol blue is another pH indicator, but the pH range is much smaller. So it's really around the neutrality. So one tips, for example, for maintaining culture is to select a pH indicator or pH indicator that have a much smaller range of changing. Okay, then you are sure you are maintaining the culture at a good pH, otherwise they will die. Okay. And another one, another tips that you can use, for example, when you work with ammonium oxidizer, it's the buffer. This APIS buffer, it's quite a neutral buffer, and it can really help to maintain the cultivation, the culture. For preserving bacteria, heterotrophic bacteria and fungi, you use glycerol, and they will show you how to do. But for example, for preserving ammonium oxidizer, you cannot use glycerol. You have to do, it has been shown that it's much better to maintain chemolithotroph with the medium with a percentage of 5% of the MFO. Okay? This is much better because it really avoid the cell to be frozen, and then it really helps for cultivation. Another thing you can use to maintain as soon as you isolate chemolithotroph is subculturing, continuous subculturing. But of course, this as in the collection require a person who is really dedicated to this, like Lucas, I think, is doing. So it's not, and the other thing is the continuous culturing. So, but of course, for this you really have a purpose, a meaning, and the money to do this. This is a small parenthesis, and I give again the floor to the fungi.
Okay.
Facciamo interattivo.
Yeah.
Okay.
It's like when, as I told you yesterday, the idea here is to basically have tips and tricks for cultivation from different experts. So we are trying to give you the hints to like personal experiences on how we do stuff when we want to preserve our microorganisms. I've been working in the culture collection at the University of Turin for several years in the past. And I was the curator of the marine culture collection section. So what I want to give you today is an overview on how to preserve fungal strains Saccharomyces for long term in a culture collection. If you want to build your culture collection, besides these tips and tricks, for ParAqua, I already gave a lecture that is available on YouTube on for example, legal framework, biosecurity level, what you need to get the quality standard that we were talking yesterday with Christine. So if you want to build the culture collection with all the data, all the procedure, all the methods, you can look at this video. So I will not be redundant for the people that already listened to this presentation. And also with the material that I will provide you. So if you want to build the biobank, I will give you as tip or tricks an Excel file that we use for people in our group to deposit the strains in our culture collection. So me, Sele, I manage the collection together with Esther that is dealing with the bacteria culture collection. We have the curator of the fungal collection that is Selene, that is not here today, but she has done an amazing job in developing this kind of files that you will have. And we also have Pietro and the guys. So what you will need to build your culture collection, I'm just showing you basically you will need to have associated to all your strains data that characterize these strains. So this is a very valuable information that you need to keep. So you can build your strain catalog. I will give you the explanation, including mandatory fields for that minimum data sets that you need, for example, for mere standards. No? So you will have a catalog. And now you can all access to mine. So this is our catalog. So you will see for each strain, we have a lot of information associated, methods for preservation that you will see also today, cryopreservation, for example. We have the medium specific for this kind of strain. As you have seen today, some microorganisms need specific medium. Some of those are in the file that I will give you. No? So I will give you this empty file. So you can fill it with information from your culture collection. These are medium that we are using for our internal purposes, but you can add yours and change, no? And also, if you want to structure a culture collection, you need to and make it internationally recognized. You need to have the legal framework for this culture to exist, because you know there is this Nagoya Protocol that you will learn about tomorrow that has some constraints. If you go in some country, sample a microorganism and keep it back to your collection. So you will learn more tomorrow. So I will give you this empty Excel file, and then other form that we can see later in case. So if you want to build your collection, you have an accession form for people to deposit strain in your collection. So if some people say, hey, look, I want to deposit my cyanobacteria in your collection, you can make him fill this form. So you have all the minimum dataset information, as well as 2 documents that you will learn about tomorrow. So I will give you the documents that we are using in our collection, and you will know the importance tomorrow. So basically what I want to give you as a practical today is just how to do tubes to preserve your fungal collection. So for fungi, this is less of a problem than for bacteria, because bacteria, if you keep it alive, they lose a lot of genetic information in a very short time. So if they have plasmids or specific genes, if you maintain them in culture like this, you will lose it probably. Some of you have ever done tubes? Milos, no? Okay. So you can help in case if I need help. So this is basically will be the practical part. Why I use tubes instead of plates? Because of these organisms here that are mites. These are specific. This is a species of mites that really like icomar seeds. They go and crawl in your plate and they eat the fungus. And what they do is they poo from one plate to another. So you will have cross-contamination among your plates, in particular if you keep them in the long term. No? And also plates become dry very fast. So this is the method that we use in fungal culture collection to preserve icomar. And Pietro also added here some information about the sustainability of having fungi in the cheese. So since we cannot use the Bunsen here, I made a video. I work with no gloves because I don't want the gloves to melt on my hands, but of course I'm working with the non-harmful fungi. This is how it works. You will have this video, and then you will do it directly. You will find. So what they do is to sterilize the loops. Maybe we can do it in groups, no?
Yes, immediately,
Okay,
straight
great.
away.
So maybe we can learn by group. So now we divide
Yes.
in two groups.
Yes.
One group will do bacteria, the other one will look at the boring video, and then make the practical stuff. Okay. Then I just brought some of the plates of common contaminants, airborne contaminants that I usually see in the pictures in the ParAqua chat. So you can then make observation at the cereal, and also you can do slides at the optical microscope if we have time at the end. So because I not a lot of time, I see pictures in the chat, so this might help you in some way.
Okay.
So we divide it into groups, pointing to the fungi and the bacteria. For a participant, I will stop the recording. Right. Okay, we will start the recording
We
once
do
we
the
have the videos. Okay. Milos, since okay, Milos. Pietro went with other videos, maybe that and he will also can help me with the explain how to make the change. Okay. Just need also psychological support. Ma non possono fare quindi facciamo
Come devo fare?
Look, this is one part of our culture collection. So this is how it will look like. So this is printed up formally in our culture collection. Each tube contains an acrylic of a specific strain. Okay, microvirus is that. I will not. So you can see these are mostly like marine extract. This is the particular. Yeah. Nectar is the most common that you can find in marine environment. Nectar and menta, that now it was.
One species.
Yeah.
It's maybe that's one very recent sample. But it's not the nectar yet, but they're nectar. The asexual state is really hard to understand.
It makes those like start like sports, right? I'm sorry.
Yeah, we will show.
So
Not nectar, nectar
when you kind of up and you put it in the fridge, they last 2 to 3 years.
Really? Wow.
I will tell you that. So 2 to 3 years in the fridge at room temperature. It depends on the season, a few months. Okay. And you can see You have the database, sorry.
No, I think it's better because we are not so many. So we can watch, everybody can watch, you explain once, and then we move to the.
Yeah.
But because
But we are not too many people, so.
right, so each one of our string has the codes, and you will see in the Excel file. So basically, when our cultures are in the final form, they have the matching codes. When all the metadata are okay, they are in a final codes, and so we keep this. And in the data set also we write down when the string needs to be transplanted, and you will learn how to do it. Why using these cotton cups and not the silicone cups or the metal cups? Metal cups are subjected to contamination. Silicone cups can dry in time, since this can be taken for a lot of years. And if silicone cups dry, then the head goes inside, and then your culture is contaminated. And this cotton assure you that they can reach the go inside, but not mice, not other compounds. You can do this kind of cups even with normal cotton. But what I use,
Right?
yeah. What I use,
For this type of things.
so this is the thing. This is hydrophobic and not hydrophilic. So hydrophobic, so you can all heat plate and it will stay. Aha!
Only new.
That's why I call it. So the first thing that you will learn is to do the cups that you can also use for like these cups can since you
Flat.
homemade it, you can use it even for
And my flat.
for different types. So the first part that you will learn is how to make it. Group
Yes.
can make.
Yes, yes.
Then I will show you how to.
You show one for everybody, then they can try.
But
What do you think?
Yeah, but you will see how to create. So the first thing is to know how to do the cups, so you can have students make them.
Okay.
Sorry, it's not that your student, but it will be the place that makes you use the cups because it's super boring. So with teaching purposes, they will make you, and you will be the place to play. So basically, you can use what illumination.
Yes.
You can show. Because I'm a little rusty.
I wasn't trust, so I
Okay.
give this to my students.
So give it.
But
Yeah.
basically, you just
So
Everybody has students, that sounds nice, like.
So you make a piece like this, more or less.
let's try
You
to.
close it on all sides.
No, we missed the technique of,
Okay.
Miller.
So just try to squeeze it inside. Some
So that's the difference.
That's
Strongness and,
Yeah.
technique. And
everything.
so
Different
you can make like a nice haircut if you want, but it's not compulsory.
So my technique is more or less I make one drive and then
Here we can see your hands.
so more or less I do the first and then okay. So I cut the cotton like this. I close it on the fourth side. And then I make like this. And then you need to push it inside. Like this. You will learn how to do that. I broke a lot. So the secret is it does not have to be super puffy and because it cuts and it's okay. But since you will use the flame, it is better to have it at this dimension because when you will use it at the flame, it will not burn. Okay? And the other important thing is that you can need to do like this. Okay?
You'll be sure you can take it from the cup
If
without.
it's too lazy, okay. Not lazy, I would say.
Use.
Loose. It's like this. Microorganisms can go inside, and you can even lose it when you have to use. So this is the cup, and it's very nice. And it's okay. You will do yours, and you can take it home. The one of the plastic tubes you can take home on the plate.
When Laura got married, there was a bouquet of flowers.
Laura, can I ask you one question? Because for instance, in my lab, we don't just use the cotton. Usually we have an outer layer of gauze. Do you gauze? These things that you use to
Like for
clean.
bandages.
But
yes.
The cotton. Gas.
Yes, you also use gas because we also make this and then put in the gas and
Yes, I was.
sew it. So
Well,
just,
I usually put a lot on top, so but I have it so that we don't have all this little fluffy cotton coming outside.
It's cool because it's, as I said, we have all different.
Yeah, I don't know if I need actually to put in the box that, but if we have some, maybe tomorrow we also show how we do
Sure.
ours with that.
That would.
Then you have to, because it's just cotton based also. So, yeah.
You can use chicken sticks, you can use I don't feel it. Look, I have a flask here. So for example, if you want to make it, if you want to make like cultures and on the shaker, you can make it bigger, but it's the same. So the dimension, then you fill, and then, okay. So then if you want to autoclave the plant, you cover it with aluminium foil. Okay, I don't have enough aluminium, but you can cover all and then go to autoclave with your medium. So this also is the trick. Okay, try. You have a lot of one group has the so you can try make your own.
So Lucas, after,
So please take some cotton and do it.
Lucas, after you prepare it, then you autoclave it, and then sometimes, for example, we put in the dryer
Okay.
to remove the humidity from the oven.
100 degrees or more?
No. 60 for a while, before, 60 degrees before, because otherwise the medium job is still at very high temperature.
Okay.
So then you have to have 60, maximum 60, and then of course you can, so it depends, if you have to do a solid medium, then you have to be careful with the agar.
Yeah, of course.
So,
So
When they are doing this and learning, you can do it.
io non devo solo fare vedere.
So make it and you have to choose, okay? Too much, okay. I was always thinking that it's like this, it's 4. That's more or less the one you did for like. Close.
If you will close,
They eventually die.
it's like the buttons of wine, horizontal position.
Either we have them in place when we are working with the cultures, or then for us, we usually see them in cultures. We will show them.
sorry. I have one expression. Is never sounds like. Yeah. It's like for an expression. Very important. Yeah.
No, but I usually use the cotton. Yes, but
It's perfect. You need some cotton to go inside, not super fluffy outside. Yeah.
You're hired.
But if you make it with this plastic, you can bring home. But just so try with the plastic and then you can keep it in your luggage and bring it home.
No, but I was telling that, for instance, in our collection, we cannot even put your plate. We either have any plate, we are in the next place, or then just put it in the back of my plate. Then I probably have to. Yeah, I'll try.
Yeah, so this is usually my tip. If you can hold it, yeah, if you can hold it like this, you can hold it.
Without the customer.
No, even without even if you have a little bit of you can hold it like this, it's tight enough that you will not lose the cafe. And usually the tight on the spaghetti, you can hold it. Perfect, Anna. If you do it with the plastic, then you can put it home. This is vicious.
In my, I don't have experience with paraffin oil. I no, paraffin is yeah, that we use. That we use, of course. Then, yeah. But that's the thing. So if you keep them for very long, you really need to have a very clean plate, because otherwise I might. I have lost many times cultures. I will give it tomorrow.
Before, when I was talking about the medium,
This is really perfect. What to be done with the plastic tubes?
I have done 50,000 even my, you have to.
the rest of that's it. And one trick to the kind of the time where is to use
Yeah.
100% concentration. And then, but the rest of these are your better line.
If you make
Kind of not bigger one.
Intellectual balance, rather.
the one piece
Twice more. But the ones that keep your
That's useful.
Here's like a cotton god.
Yeah, but the next one didn't put it, so I did like what it was. Then I wrote from 25 to put 1,000 to suit.
And also like, so you see this with the meters, because it's for the medical things, so I divide them in like meters and meters. And we chop them in the right side, and we put it in the outer part, and then I make some little more, that's what I want to do. And I reuse them, so if you re-autoclave, it's fine, then it's very great. You have two methods to actually do that. Plastic, you just have the glass empty. There's a lot of for instance, what we do is sometimes we have open sterilization, like glass, the open sterilization. Moment of attention, please. If you have done your plastic tubes to bring home, can you also make some cups for it that already have the inside? Don't make it like this. It's just that we will need it later and I don't know what to do.
This is
we have options.
So
So they have the so wait, sterilization, we can reduce them and many, yeah, but I mean, the content does not burn. So you can actually reactivate multiple times. If you try to try sterilization in an open, and the cotton and the gauze starts to be really fragile. If you do exactly cycles, only two is the maximum. Starts to be very dry and fragile, and then it comes under you. But without play, I
Right.
but another way. And
Oh.
and they are, but the other one, the other group. We have another group.
Ja, to pani podkładaj potem. Na pewno się ogarnie, potem powiedzieć,
No, io infatti delle solite bottiglie, ma per il fino per il sistema. Ma te mi viene su un plate che
Yes.
Yeah.
What do you think about this type of cups?
Yeah.
I have difficulty with this.
Okay.
So
So
they
now,
don't stay
when
so
we
much
make
for, I think for algae are fantastic.
Yes.
But for bacteria,
which I
for bacteria, the best are not even this, but they're really tough.
both
Yeah, they're really tough.
Oh, wait. Not
Bacteria. So they need some air,
You're bringing home,
but
but
they
guess
cannot
what?
be, and then of course,
Okay.
if you use this, all this is organic. So then your bacteria will stop growing. Because you have organic, everything is glass and metal. And for the medium, you use this. And the stock, which this is just, of course, the bottle. We have stock, this kind of bottle with silicone cap. We have this kind of cap. All the media for lithotrophic bacteria are prepared in bottle like this. You sterilize them, you close them, and then you have stock solution. So 100 per 1,000 of the element, the trace element solution is 1,000.
Yeah, I do the same. Yeah.
Exactly. And then I use needle. Keep the needle in always, every time the syringe. So then you will never open this again.
Okay.
So never try with gloves, just remove the syringe and put the amount you want. That's the trick. So we were the lady from the NEO, the institute where she was working in the Netherlands. She was really maintaining this huge. They made us hate. And we have some species that's really hard. Now, for example, they disappear. So if deposited in culture selection, you only two species. They are lost. And she was using this technique of the needle. I find it very useful.
Okay.
But then you will never remove it.
Okay, but you will never, you will not get contamination from the people.
Yeah. Maintain and before then you have to check the culture with so when you subculture, one portion you subculture, so you put in a new medium, and the other one you put in an organic medium.
Okay, then
Growth of
you go
otherwise
from the
and you will never be able to recover an ammonium oxidizing cultivation that is being contaminated.
you prepare.
They will outcompete and they will. If no, they will use the yes, exactly. That's why nobody wants to work with ammonium oxidizing anymore. It's too hard. Like for maintaining the culture, but the point is that as soon as they use light, somehow it's easier, but as soon as you have to consume the medium, because then they consume the medium, then you have to provide your medium. So this subculture is challenging. Every week you have to change medium.
What amount of medium final which you can obtain from this?
So the trace element solution are 1,000. So if I want to make 1,000 liters, or 1 liter, then I use 1 milliliter.
So make the stage. But I use sometimes 5 liters per week. So this is for 1 week.
Yes.
Because I use.
So what you do there?
But you use it yourself. Anybody is taking care of the collection?
Usually.
So
Do you have somebody else who is helping with the collection?
One guy helped me, but they also work with different institutes, so that's really.
Sorry. No.
It's quite complicated.
So. You make all the cup, okay, all the flat. And basically it's 15 ml for each tube, more or less, depends on the dimension, but you can make one trial and then you can test the medium. Also, you can prepare the medium, make the tube and then select the entire medium. Okay. So what you do is you select your own favorite medium, PDF, whatever suits you with agarized medium. And what you do is you dissolve the medium before on a heating source. It can be this. I used to do it bendery, like with the pots full of water with the, and also I used in the past big, that when it gets more or less like transparent and it's not how to say
Turbid.
it's not turbid, it becomes clear, so it means your agar has dissolved. Then you pick up everything. This is also one thing that you should hear if your cup is correct. So this is not correct. This is not correct.
We are bad. That was fun.
So, but you can try, there are different methods. We have an automatic dispenser, but you can also use basically, how to say, baker. Or it is good also to have this current, and you put the medium, the liquid medium inside, and you can try it. You can try.
You can try to use the multiple dispenser.
Also, like everything goes, the important thing is try to keep this part clean, because then you will, if the cup gets wet with the medium, then when it solidifies, it comes also dry, and so it does not stick well with the cup. So, so basically, you take the medium and put it to have it's usually up to yes. So since this will be hot, then so this will be hot medium. So it is better to leave, it will be the same heat. So leave the tubes there and then just dispense. You can leave a lot because of space like this. So then it would be 45 degrees.
Okay.
Because ecco di clarino in English. Clarinet peak. It will solidify.
Angles.
Yeah, it will solidify. Okay, you can try. Because this is how they will solidify. So you don't feel up to the top.
That's the name.
More or less.
Fine.
A little bit. little less than a cup. Because then we need to dry them like this. So basically, you do the cups before, or the students do the cups. Then you remove the cups and you put the liquid medium. Okay? It is too much. No, but you can just pour it in the no, like not. It always happens.
People into the peristaltic pumps.
Yeah. If you use a peristaltic pump, then be careful to prepare an amount of boiling water to clean the tubes, because if they are solidified, then
They can be solid.
so once you prepare everything, you then pass the hot water.
Just that, because you're saying before I even fully, very fully. So we are saying about the dissolving in the agar, and so this is not just autoclaving the medium.
No, you don't. You have to play later in the tubes. So you just boil to dissolve. Okay. So you just mix that and heat so that all the medium is dissolved and it's not so fun.
The tubes go to the autoclave, and then when they come out, it's with the mantle, and then they solidify.
Okay, we can.
And then you have the melt twice.
Then what we do when you will all prepare. Already heat it. Just close again. One for each, because we need to close them. So usually I make the cup and I keep the cups in the same order as the tubes, so nothing say dimension, because usually you have tubes of people. Okay, so if everyone try this, okay? Yeah. What is the name of your phone? You close it again. And what you do is to use this kind of whatever you have that goes in out of place. But
Generally, I use glass speakers because I don't have this fancy thing.
yeah, but the glass speaker are in a little places which heat it.
And they heat, and also the point of glass beaker, so open things and not no holes that they retain water into. So then you have your tubes, and then you have the glass, and then you have some water in. And so if you're not quick to remove, then you have all this water, kind of this water, so it could be old, and the water from the autoclave should be sterilized by so it's better to have something at least with old.
You do it before the cups, because usually it takes the amount of work, like the amount of soup that you make, that you can keep it in the fridge. Then they are close, they are sterile. Usually when you do the soup, then like this one. You make big amounts, so there is become a big amount. So you prepare the cup the day plus the day before. So the important thing when you put it in your autoclave is to not turn the tubes. So they need to stay upside down. The upper balance. So you can just put something cotton, or if you have aluminum foil, just to keep them straight. Yeah. So usually it's full, then a double layer aluminum foil, because it gets wet. It resistance to water, but it does get wet, no? Like I apply to my medium. So, and then it added, you don't want to make me the fake autoclave. Okay, let's make. So we make the tube. So we cup them again, we cover, and then autoclave. So this is the 120 degrees for 20 minutes, no? So they came out. Of course, I'm wearing gloves because they are incredibly hot.
now it's not. But now I know.
It is really well.
While they are still hot, so the medium is still liquid after autoclave. You have all the different type of films to have this. Okay, it depends. Okay, this kind of you try them like this. Okay, just put them like this. And it's better to give it to leave it overnight, but depending on the season in winter, they solidify the up.
Like mine. Keep it sideways.
And we have it ready.
Exhibition cooking show.
So at the end, they will look like this. Okay. So in that tube, then you can put your focus parts, and it will stay like this. So it has its female. Even if it's an actress,
Actually, they have male and females.
I don't know it.
Male.
But like the fungus will get the spread, so it has the space to grow and to use the medium. I will show you then how to inoculate and transplant. Just want to let you know. So good and bad.
Another.
Yeah.
You leave it like this so that there's more surface area
Yeah.
for it to grow on.
Yes. And
If you left it like
so when you will inoculate your fungus, then you will need how when your fungus is growing and it becomes like this by your tube, now it's time for you to transplant, and you will see how. If it becomes like this, you have been too late. But fungi can really survive. We just put the sterile physiological water,
Physiological water.
or sterile water, of course not hot. And then you leave it overnight, so you rehydrate, and then you can try. And some fungi really can survive. And the other thing is when I want to not that my fungi will go in connection, but I have some tubes. So this is I want to clean this because I want to reduce the tube. So with the microorganisms, even if it's not pathogenic, then it's always a good thing to not touch a living microorganism. So what I do is I put the water, or if it's like this, I don't put anything, and I put it back in the autoclave. When it's autoclave, then you can throw away the cup and then clean it with the scolino.
Yeah, with the
The brush. Okay. Then you can use the tube again.
yes.
Okay. So how do you inoculate it from? It is not good to use the tubes for isolation because in tubes it's more difficult than the plates to see trap contaminants. But when you get the autoclave cycle is the depth of bacteria. It's 20 minutes for well, so, yeah,
It's very high.
you can use even longer cycle. It's
depends on medium, I guess. So some medium require high temperature or long time, but usually
For bacteria cannot be applied.
Good.
Just boil it. It's kind of, like for example, with chromogenic medium, is chromophore, is very sensitive, so you cannot even apply. So you use no. Marinaga, yes, but for example, the chromogenic medium is the fine medium are cannot be applied.
And there's no. They losing the grabbing the solid after.
Yes. Yeah, that can make it. That's for chemical stuff. Yeah. There are tips and tricks that you learn. But for example, with this, so you cannot always autoclave medium. Like or other blood for clinical, for example, you cannot autoclave because then you will destroy the blood cells. That it's so every time as soon as if you get to become by working, you get really able to be as clean as possible. This attitude in the lab will help you and change also organisms you are working on. So the bigger you are, it's like an attitude in life. You don't know how to say. Yes, it will help.
Yes. Yeah, you will have this. I don't know, like many of you are working this, but this ability to work on the laminar flow and not pass over the stuff and that all this kind of.
Yeah, similar situation in the diagram where we added the vitamins. And in the original percipit, yes, information that we should add half milliliter after autoclaving.
Yes.
But my friend from Szczecin, because she was working with marine diatoms, told me that, we add double volume, but autoclaving and so. So not half milliliter, but 1 milliliter and autoclaving.
It's good to do the different experiences.
Yeah, that's exactly.
More than
Also, since, for example, here, this I told you before, to maintain this stock solution, you can have here, you can also put the 0.22 filter.
yeah.
And then every time you take, you regain, let's say, filter sterilize the solution. So if you can have it.
Also, yeah.
So how to inoculate from plates that are clean. So this is the clean microorganism. This is the clean alveolarian plants, and it's basically an airborne function as you breathe. But they basis. Like fungi testing. It's since a contamination, but it's an overgrowth of microorganisms. So black is coarse and white is mycelium overgrowth. This is the clean plate. So you are done your isolation that was not the of this course. You have done your isolation out, so you got from here to the tubes. That method is use the microbial loops that for fungi is like you just take some mycelium plasts and then you put it in there. And you can modify microbial loops using this kind of wires that can be sterilized with flame. Or if you don't have this and you want a cheaper version, then you can use insulin syringe. I use this also to isolate the microbial. I just all up. So I have this, so this can pass through my tube, and then I use it. I take the mycelium, I sterilize the bones, and you can sterilize the tips very quickly. The outside, that's why it's preferable. Yeah, so you just like this. You take and you can put in the tube. I don't know, because I worked in ParAqua-COST in the plant pathology lab. They use very good techniques. They were using no molecular biology, nothing, and they with molecular. So how do you transplant or isolate? I will show you the video so you can see that. So the best thing is you can use a tube holder, or you can use a support that does not suffer from the heat to put your tubes there. So, as I told you, one thing, when it goes out from autoclave, it can happen that the cotton sticks to the tubes. So best thing before to start isolate or transplant, you do this and you turn
Unlock.
so the cotton will not stick on the tubes and not get in the way. So you can try this out in autoclave. So the best thing to hold the cup up like this. Okay. So before to start, rotate the cup. So to remove the cotton. So that's why this fungal loop can be really sterilized very, very well at the flame. It is good to sterilize all the part that goes into your tube And as you know, the Bunsen flame, the hotter part is the outside of the flame. So try to be tangent to the flame so your loop becomes red. Each time you open the tube, you sterilize at the Bunsen. Okay. You take some of the mycelium, sterilize, close. Take your cup, sterilize, put in the new tube your fungus, sterilize, close, and then you soon sterilize the loop so you don't spread the microorganism.
The next.
So you have here, you go inside, you take some of the mycelium, very few, it's okay. Even for sporulating fungi, really a small amount is okay. If you're not comfortable with taking 2 tubes that we will try later, you can just put it down, take the new tube, put it down. The important thing is you sterilize the top first. And you see it becomes really red. It should become totally red, okay? I don't know what else it is. So really, it should become this red. That's why it's good to have a backup wire. So when it becomes.
In the second one, the purpose was to show the amount of material you have.
But if you have sporulating like penicillium or aspergillus, just touch and it's okay. And this is a video homemade by Zach and me this morning. Don't try this though. What happens if your cup will take fire? So 3 methods.
No.
1.
Wow.
3. So, but in this case, you will lose the culture because it gets contaminated. So it's good to have a lab that is full cotton. So if you do like this, you don't take it. And you need to be very brave. So when I have students, at first braving, but so it's a torch. And also because if it takes fire and you don't extinguish very well, you put it back like here, and then you put it in. It happens to me, so don't worry. And yeah, this was Generalis medium, and this is a surprise picture from my.
Yes.
But this is not here.
We are here in the same institute, 2009. And on the right, Serena, taking course in 2009 here.
Okay. So do you want to make some bacteria, and who wants to stay and practice tubes isolation?
Yeah, I think that will show if you want to try just to spread, if you never tried to put on lighter plates to see how good you are, how gentle you are, not to make some stripes on the other plate. So if you want to.
So with the going.
Yeah, or you can also use mine. Mine is a very one.
Yeah.
It's just
I can show single color.
exactly.
Just like
Huh? You have
so can I just say one?
yes. The last thing is cryopreservation.
Okay. No, cryopreservation. That's what we do for fungi. If we keep a plate with glycerol, 20%, I can share with you. So 20% glycerol in the video. We cover with sterile glycerol, the pure the actual culture. And then we use, I don't know where I put it. I will show you. The size. Well, I will tell you later. We have capillary tubes that are sterilized. Okay. We have the problem with the bone medium. We put it so this capillary tube will be full of your fungus, the cover with glycerol, and then we put in few vials. And the best thing is you can put it in -18. You can first, the best thing is to fast decrease, fast freeze, and then slow decrease. That way is -152. If you don't have -80, it's okay. And if you don't have any of this and you want to long preserve, you don't have a fridge, you don't have cryotubes, you don't have whatever. This is another tip or tricks from the pathology lab. This is the tube that you have prepared. You prepare, what is the plan? You sterilize the glycerol and you cover your culture with glycerol. These are tubes from 2012 and they're still alive. Not all the fungi can stay like this, but a lot of fungi remains alive under glycerol. So you can keep it room temperature, just cover in glycerol, mushroom under oil.
Yeah.
And this is still alive. So when I need it, I just take and make fresh culture. And of course, you make fresh culture and you can cover with glycerol the new culture.
And how do you get to it? Do you just stab through the glycerol?
With the, yes, with the loop.
Okay.
And the important thing is when you use the loop for this kind of purpose, you need to sterilize very well. This part, because oil stays in skin. So, and that's it. That's all I need.
So they go inside and then you put it like that. You see that they grow, they already stopping,
They stop.
completely stop. Okay.
Super silly question. Is it fungi, fungi?
Nobody.
Everybody
several meetings.
It's bigger.
Okay.
And I would say it's
All of them are good.
Okay, how do you think?
Fungi.
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 Okay.
Okay, but they are correct because one is the Latin pronunciation.
Okay.
One is the English one.
What is the last?
Fungi. Fungi.
Kind of.
Fungi. Fungi.
No, fungi.
Fungi.
Yes, because the H